Good afternoon, everyone.
Good afternoon, madam.
Okay, so shall I start?
Yes, madam. You're ready.
Okay. Good afternoon to one and all. Good afternoon, today's resource person, Professor Christopher sir. And good afternoon to Vice Satnaran sir, Principal, Government Degree College for Women, Jagityal. And today's convener of this session, Dr. A. Rajni ma'am, Head Department of Botany, Reddy Women's College, and Padala Tirupati sir of Department of Botany, Jagityal, and all my dear students. Today we gather over here for an extension lecture on applications of rDNA technology by our resource person, Christopher sir. I welcome you once again to all. Now I would like to invite Dr. P. Manjula, Assistant Professor, Department of Botany, RBVRR Women's College, to introduce today's speaker. Over to you, Manjula.
Okay. Thank you, madam. Good afternoon, one and all. So it gives me an immense pleasure to introduce today's resource person, Professor Christopher Reuben, sir. Sir completed MSc and PhD from Kakatiya University and awarded University Research Fellowship at Kakatiya University, Warangal, and Senior Research Fellowship at Kakatiya University, Warangal, and sir awarded Research Associateship at University of Delhi, South Campus, New Delhi. Sir held different prestigious positions like course coordinator medical transcription, Kakatiya University, additional controller of examination at Kakatiya University, and head department of botany, Kakatiya University, and director foreign relations officer at Kakatiya University. And sir presently working as a dean, faculty of science and research development, Chaitanya deemed to be University. Sir has 16 years of teaching experience and 22 years of research experience. Sir area of research are plant tissue culture, genetic transformation, and molecular markers. Sir has 48 research publications and sir awarded under 8 students at PhD under SAR's supervision, and 8 students are currently working under SAR's supervision. Sir attended 25 national and international conferences and published 8 book chapters. And Sir has a resource person for 20 conferences and sir given the lectures on 25 invited lectures, and sir has a membership of academic bodies, 20 memberships, 20 academic memberships. So through this brief introduction, now I invite sir. Thank you, sir.
Thank you, Manjula. Now I welcome Satnaran sir, Honorable Principal of Government Degree College for Women, Jagityal, to speak few words on this occasion. I welcome you, sir.
Okay, welcome. Thank you, madam. Hello. Good afternoon to all.
Good afternoon, sir.
Good afternoon, sir.
Good afternoon. Our life science department is sharing the knowledge online through webinars and extension lectures. Today, our botany department is organizing applications of rDNA technology with the RBVR college and our college combinedly. And welcome to Christopher Garu, Professor Christopher Garu.
Thank you, sir.
Thank you, sir. And Dr. Achutadevi Madam Garu, Principal, RBVR College, and conveners are Dr. Padala Tirupati sir and Rajni Devi Garu, and our college staff members and other RBVR college staff members. Welcome, sir. Welcome, sir.
Thank you very much, sir.
Thank you, madam.
Our principal, ma'am, Dr. G.H.R. ma'am, is busy with her tight schedule, sir. Today we are having Women's Day celebrations at college, and madam is held up over there in that occasion, sir.
1
Okay,
minute,
madam.
madam.
No problem.
Women's
No
Day,
problem.
advance Happy Women's Day, madam.
Yes, sir. Yes, sir. Thank you so much, sir. Thank you, Satnaran, sir, for accepting our invitation and to be here, for encouraging in all endeavors of continuously encouraging our students and us for conducting all these events along with your college. Thank you very much, Satnaran, sir.
Okay, madam. Thank you.
Right. Thank you, sir. Sir, now I hand over the session to today's resource person, Christopher, sir, to enlighten our students with your immense knowledge on biotechnology. Over to you, sir.
Thank you, madam. Thank you so much. Can I use the PPT?
Yes, sir. Yes, sir. Okay. You can share your screen, sir.
Post disabled participant screen sharing.
Yes, sir. Just a minute, sir. I'll do it.
Transformant. Once the putative transformant is produced, it is transferred to a greenhouse for acclimatization. After acclimatization, we transfer it to a field condition where we call it a T1 plant, that is transgenic plant 1. And then this is not it. So what we do is we need to confirm whether the gene that we have inserted from this site to this site has really got incorporated, got integrated into the genome or not. So that has to be tested out by using this technique called Southern blot technique or South blotting technique. And also we have bioassay where we use screenable markers, that is GUS or green fluorescent protein. Once this is confirmed, then we carry it to the second generation, then to the third generation. So at the end of third generation, we confirm that the plant that is obtained by this method is a transgenic plant. And once it is confirmed as a transgenic plant, this will be transferred to a farmer's field through a seed company. This is what happens in case of rDNA technology with crop plants. So these are very important 7 steps that we need to follow and understand, right? Okay. What is a brief history of transgenic crops? As I've told you, all started in 1972 when the scientist Paul Berg responsible for inserting bacterial DNA into tumor virus. I repeat, he has found that a small piece of bacterial DNA can be transferred to virus, that is tumor virus. This is the first step actually. Then Herbert Boyer and Stanley Cohen developed this technique of transgenesis or transgenic plant development. And I've already shown you particle bombardment technique was developed by John C. Sanford. And then in 90s, we have edible vaccines developed. This was C. Arnzen. And then 1992, first transgenic tobacco plant, which is virus resistant, was developed in China. In 1994, we have flavor sour tomatoes. I'll tell you details of this in the coming slides. This is a company called Colgene in USA. Flavor sour tomato has long shelf life, long storage life. You can store tomatoes for more than 3 weeks. And then 2000, we have golden rice developed. This is again by Ingo Potrikis. And then in 2002, we have BT cotton introduced into India. This is by Myco, Maharashtra Hybrid Seed Company. And 2005, BT brinjal, again Myco. By 2005 and 2020, nearly 525 transgenic plants, transgenic events have taken place in 32 crops. 32 crops today have what you call as transgenics, and then they're cultivated in different parts of the world, mostly in US and Brazil and China and other parts of the world. So this is the history of transgenic crops that have been developed. These are the examples I can give you. For example, genetically modified crops, we have rice with vitamin A, call it as golden rice. Then you have tomato with a long shelf life, call it as flavor sour tomato. Then potato, it is bruising and browning resistance. This is a vegetable. Apple, browning free. Fruit, then mustard, herbicide. Sugar beet, herbicide resistance. Alpha, herbicide resistance. Soybean, herbicide resistance. Okay, what is the importance of these is cooking oil and margarine. This is for granulated sugar. Alfalfa is a fodder. Soybean is poultry feed and livestock feed. Also, we get healthy oil from this. Then cotton, this is very common these days in our country. BT cotton, it is insect resistant. Brinjal is also insect resistant. Then papaya, virus resistance. Squashes, we have virus resistance. Then mustard, again, herbicide resistance. So these are some examples that I have given you in this particular slide. So using RDNA technology and the process of genetic engineering, these crops have been engineered, and we call them as genetically engineered crops, right? So from our syllabus that is with us, we will be discussing pest resistance. Examples are BT cotton and BT brinjal, herbicide resistance, Roundup Ready soybean, then quality traits, we have flavor sour tomato and golden rice. So these 5 are present in our syllabus. I will be giving you a detailed introduction into how BT cotton, brinjal, soybean, tomato, rice have been engineered. I will be discussing one after the other. I also have given some examples other than what is present in your syllabus. That is salt tolerant rice, transgenic papaya, that is virus resistance, and then several others. At the end of the lecture, you can have a look at them. So first we'll start with BT cotton, right? One very important thing you should understand in India is only BT cotton is permitted in our country for cultivation. No other crop is released for cultivation, either herbicide or viral resistance or flavor sour tomato or golden rice. They have not been permitted to be grown in our country. In our country, only cotton is being grown. A reason for this is there are a lot of questions in GM crops. So they don't want to get into the issue of gene pollution, gene contamination. So India is not releasing GM crops other than cotton. So this is cotton, right? Okay, this is called boll. This boll is infected by a fly, the larva of this fly. Always larva will bore into the boll and eat the tender seeds inside this boll. As a result, this fiber does not grow as seeds are lost. So you can see that this is an empty shell because this larva is a voracious eater, and as a result, this larva will grow to the next instar stage. This is 1, 2, 3, 4. So this is how the larva is growing, and then finally this larva will develop into a pupa and will be released as a nymph and finally an adult. So this is what happens with cotton. As a result of this pest, infection on cotton, we lose almost 90% of the crop because of this pest. Because of this pest infection, we lose 90% of the crop. Okay, because this happens at the end of vegetative phase, and when we see there's flowering and then fruiting, this happens. Okay, so what scientists, what a farmer does is he starts to spray a lot of pesticides. One intelligent thing that this larvae do is during daytime they are buried in the soil. They come out only during night time, and the farmer sprays the pesticide during daytime and he is sleeping during night time. So when the farmer is sleeping, the larvae come out of the soil and then they make this bore and the bowl is lost. So this has a lot of impact on environment because there is no use of these synthetic pesticides in controlling these larvae. Instead, they are polluting the environment because they use excess pesticide, what is needed more than the dose. So this what happens here is these have escaped the pesticide and the loss continued still. As a result, what happened is scientists created one important strategy. What is that strategy is there is a bacteria by name Bacillus thuringiensis. The name of this bacteria is very important. Why? Because it's a natural pesticide. Bacillus thuringiensis is a natural pesticide. This bacteria produces a called Bt toxin, which is in form of a crystal. This bacteria produces a toxin, which is called Bt toxin, and this Bt toxin is crystal-like. Now, when the larva starts to consume the leaf, along with that, the Bacillus thuringiensis also, because it's a natural one, and this particular crystal will enter into the gut of this larvae. When it enters into the gut or whenever it is ingested by the larvae, this toxin, this crystal will create bore into this membrane. This is the membrane. They get inserted here. And when they get inserted here, the larva dies. This is a procedure, what happens. Okay, but the larva dies. Okay, each toxin has a what do you call a sensor, a receptor, sorry, a receptor. So a toxin binds to the receptor, and as a result, this larva dies. This is natural. This is natural. This happens naturally. This is nothing to do with genetic engineering. This happens naturally in nature. So in nature also we have biopesticides, which can be used as biopesticides. Okay. So what scientists did is they isolated gene from Bacillus thuringiensis. The name of this gene is crygene. Sorry. The name of this gene is called crygene. CRY gene, crygene. Crygene is inserted into cotton plant. So as a result of this, what happens is crygene starts to produce Bt toxin, which is a crystal. So when an insect larva starts to feed on the leaf of cotton plant or brinjal plant, what happens is they also ingest the crystal, which is called Bt toxin. Once this toxin enters into the gut of the larvae, they die because of the puncture that is created on the membrane surface of the internal membrane surface of the larvae. So as a result, scientists have cloned, scientists have created crygene, have isolated it from Bt, Bacillus thuringiensis, inserted this through genetic engineering into cotton plant, also into brinjal plant. As a result, when these insects, when these larvae try to consume cotton boll, or when they try to bore into a brinjal, then they die. This is what happens in case of Bt cotton and Bt brinjal. So also you have Bt brinjal. This is Bt brinjal. This is normal brinjal. Okay, so you have a shoot borer and fruit borer. This is a fruit borer. It bores into the fruit of the brinjal. You have 2 bores here. You have 3-4 bores here, and this is how it stays inside the brinjal. And as a result, it will consume the entire components that are present, entire content that are present inside the fruit, and this is how they appear outside. So this is not acceptable in the market because they are susceptible to this particular larvae. So scientists have engineered CRY1AC gene. I request all the students to please give some thought to this. It's called CRY1AC gene. This is called crystal 1 AC gene. There are several AC genes, so they have given it as crystal 1 AC gene, crystal 2 AC gene. There are several genes in that, but one important word that we can have for this is called crystal protein gene, and this is called CRY1 AC gene. This gene is introduced through Agrobacterium tumefaciens, into crop plants. In this case, cotton plant as well as into brinjal plant. So what happens is brinjal and cotton plant will start to produce crystal protein. So when the larva starts to eat the leaf, the fruit, they die. Okay. Now students will get one question. Now, will this crystal protein that is produced in the fruit of brinjal or in the bowl of cotton? Okay, bowl of cotton, no problem. But if it is produced in the fruit of a brinjal, whether it will cause toxicity to human. This is one reason why in our country, BT brinjal is not released. But there are other countries which are still cultivating BT brinjal. There are no cases so far. But in our country, still there is a ban on BT brinjal. Yes, everybody will get the same doubt. When it is able to affect a larvae, will it affect human system? Because brinjal is consumed, brinjal is cooked and eaten. So this is one reason why in our country this is not released, BT brinjal, and they're still under moratorium. This will not be released even in due course also. So CRY gene was introduced into both these crops, and they are now resistant. One is good under good cultivation in India, that is cotton. All the cotton that you see when you go out of your house into a countryside, into a village, all the cotton that is presently cultivated in Telangana is BT cotton. Non-BT cotton are very, very few, but most of them are BT cotton, and they have good protection against the larval infection, against the larvae. So this is how they have produced pest-resistant cotton in our country, which is under cultivation. Pest-resistant brinjal is not yet released in our country. So this is the story of BT cotton and BT brinjal. Okay, see, I've told you that. See, this is the toxin, crystal protein. Crystal protein enters into the gut of the larva. In this gut of the larva, this will create pore into the body of the insect, and the insect dies. Here it is receptor, here is epithelium. So gut membrane perforation because of the crystal. Because usually it is called endotoxin. This endotoxin becomes prototoxin. Prototoxin becomes activated toxin, and activated toxin will create a pore, a puncture, a perforation into the epithelial cell, epithelial layer. As a result, the insect larva dies. So this is the mechanism that is being used. So they are producing through genetic engineering crystal proteins, right? So this is the second example that we can see. But then the next example that we are talking is herbicide tolerant transgenic crops. Herbicide tolerant transgenic crops. See, this is one very interesting thing. Along with the crops that we grow in the farm, weeds are very common. Everybody, when we go to a field, we watch that along with the cotton plant, there are weeds also growing. Along with paddy, weeds are also growing. Along with maize, weeds are also growing. So this is already there. So a lot of weeds are there. Broad-leaved weeds are there, small-leaved weeds are there, but weeds are very common. Weeds are very common in agriculture. In any country, in our country also, it is very common. Okay, but weeds are not wanted. Why? Because weeds use up the fertilizer that we apply. Also, they use up the water that they take up the water that we provide irrigation, and they grow better than a crop plant. So what we do is we usually do manual picking, manual weeds are removed manually. This is a very common sight when you go to a countryside, to a village. Men and women are working hard in the sun, removing the weeds. This is very common. But in this case, what scientists decided is we should produce herbicides which will kill weeds but not crop plants. You should understand what I'm trying to tell you. We are engineering crop plants. When we are engineering crop plants, we are sending some gene into crop plants. But along with these crop plants, there are also weeds. So when you spray that herbicide on that particular farm, crops will survive while weeds will die. Why? Because crops have received a gene that will give them toleration, that will give them resistance to that particular herbicide. So I can show you one example. See here what happens. This is a farmer spraying lot of chemicals. They are herbicides, they are fungicides. This is how they try to kill herbs. This is how they try to kill herbs. Okay, this is what happens. Even now, nowadays in towns and cities also, we are using a lot of herbicides to control grass. Okay, this is being done everywhere, but it is also adding pollution, adding chemicals to the soil and water. They have their own bad effects or side effects, right? So this is how we use chemicals to control herbs or weeds. But here is a situation. It is called glyphosate transgenic plant. What is glyphosate? Glyphosate is a chemical which will kill crop plants, which will kill plants, sorry, weeds. If you can have a enzyme that will make the plant resistant to glyphosate, then we can be able to overcome the effect of the herbicide. So scientists found out that there is a gene called EPSP gene, enol pyruvate shikimate phosphate synthase gene. This is a gene. Okay, they have found a mutant EPSPS gene, and this EPSP gene is introduced into crop plant, let us assume cotton plant. Once this gene is introduced into cotton plant, this gene will produce an enzyme that will degrade glyphosate. This gene is present in crop plant. This gene is not present in the wheat. So when you spray glyphosate, what happens is EPSP enzyme will degrade glyphosate in crop plants, but in weeds, they die because they do not have this particular enzyme that will degrade glyphosate. So using this technique, they have been able to engineer, they have been able to protect soybean. Soybean is a very important crop in US and other places. Very, very important crop. Okay, so they have used the same technology that I've shown. They have used this glyphosate. They've introduced the gene called EPSP gene using agrobacterium tumefaciens into soybean. Soybean will now start to produce this enzyme, the chemical enzyme called EPSP enzyme. And this EPSP enzyme will dilute or will reduce glyphosate effect, but the normal wild plant, that is a weed, will die. So this is the second example, that is engineering herbicide resistance through rDNA technology. This is what happens. Glyphosate. So they spray glyphosate. Glyphosate is also called Roundup chemical. Okay. So glyphosate they spray on this particular plant and this particular plant. This is a regular plant. This is a Roundup ready plant. Okay. So when you spray Roundup, this will die, but this will not die because it has EPSPS gene. Okay. Glyphosate herbicide kills plants by blocking the EPSP enzyme, an enzyme involved in the biosynthesis of aromatic amino acid, vitamins, and many secondary metabolites. As a result, what happens is because this is not having EPSP gene, it dies as a weed, but this is having EPSP, so it survives. This is how we have developed herbicide resistance. Okay, and then using this technology, a lot of other chemicals have been produced, like glyphosate, Roundup, Biolofas, bromoxynil, glyphosate, and sulfonylurea. All these things have been developed, and now these are sprayed. In countries like US, where one farmer will have about 300 acres of land, 200 acres of land, this is very important for them. So they use, they spray herbicides, and then the crop plants are protected, while herbs and weeds, they die, which do not have EPSP gene. So this is one another important area where rDNA technology through genetic engineering is successfully used. Then the next example is, this is called flavor sour tomato. Flavor sour tomato, right? In our house, we purchase a tomato from the market, right? And when we purchase this tomato from the market, it is fresh and ripened. But when you get it to the house, maybe after a weekdays or so, you get fungal infection. Okay, this is fungal infection, and then the fruit starts to rotten. Okay, so hardly 7 days. Okay, hardly 7 days this particular fruit can survive. In summer, not even 7 days, 3 days, 4 days, tomato fruit will be rotten, right? So once it is over-ripened, over-ripening is due to softening of the skin of this fruit, and the softening of the skin is done because of polygalactose, because of polygalactase. This is a polygalactase enzyme, that is PG, call it as polygalactase enzyme is produced, and polygalactase enzyme is produced. What happens is the skin or the outer coat of the fruit starts to soften, and once it is softened, then easily bacteria, fungus can enter into tomato, and finally it is rotten. So what scientists done is, they have designed this particular technique. What is the technique is, this is normal tomato. A normal tomato will produce PG, polygalacturase. Okay, this is a normal PG. Okay, this is produced by normal mRNA. This is sense RNA, and this is a protein that is produced here, and then naturally rotting here. Okay, takes place here after weekdays or so. What scientists did is, suppose this is a normal mRNA, sense RNA. Scientists have made antisense mRNA for PG gene. Okay, this is sense RNA. You also have antisense RNA. So they have made this antisense mRNA. Antisense mRNA is exactly complementary to the sense mRNA. So what happens now? This mRNA goes and joins with sense mRNA and will not allow this double-stranded PG gene to produce PG enzyme. PG enzyme is not produced. Why? Because this is it's not single. This is complementary. So they bind, and as a result, PG is not produced. So this is called antisense RNA technology. So they have used antisense RNA technology, and they have produced a synthetic antisense gene, and this synthetic antisense gene goes and joins with the sense mRNA. As a result, PG, polygalacturase is not produced, and you see that this fruit will not ripen or will not rotten. You can store this in shelf for 3 weeks. Here it was just hardly weekdays. In summer, 2-3 days. But this fruit, after this technology in flavor sour tomato was worked out, then they could store it for 3 weeks outside a refrigerator on the table. It can be stored for 3 weeks. All this is because of antisense RNA technology, which is being used for flavor sour tomato, for a long shelf life of tomato, right? This is the fourth one. Then the next one is golden rice. Why are we interested in golden rice? Why we want to produce more rice, which is having vitamin A? Why? Because children suffer. I think I have a statistics, right? Yeah, statistics. Children suffer from night blindness, from xerophthalmia. Okay, and then they have the general childhood diseases and infections also. All this is because they don't have vitamin A sufficiently in the diet that they take. Okay, as a result of this, 124 million children are deficit of vitamin A. Annually, children of the age group of 1 to 4 years, about 1 to 2 million children die every year. And 5 to 10 years, 0.25 to 0.5 million deaths in children occur. In 2005, 119 million children and 19 million pregnant women in so many countries were affected by non-availability of vitamin, we call vitamin A deficiency. So what scientists decided here is we can insert the gene for vitamin A into rice plant. See, one very important thing that we can understand that how people are concerned for protecting children and also pregnant women is this man is from Sweden. Swedish country does not produce rice. They don't grow rice. They grow wheat. But this scientist who was very kind, what he did is this rice could save 1 million kids a year. They have designed rice, and this rice now, the grain of this rice now will start to produce vitamin A. One thing also you should remember, animals and human cannot synthesize vitamins. We have to get vitamins from plants. We have to get vitamins from bacteria, but not from animal source. So this man and their group finally were able to produce these rice variety. We call them as golden rice. See, this is the normal rice that we consume. We call it as wild type. Then they have inserted a gene for vitamin A called golden rice 1. And now you see it's darker. It is golden rice 2. I came to know that golden rice 3 is also available now. So see the color gradation here. It is pure transparent. It is slightly yellow in color, and this is gold in color. That is why they call it as golden rice. Okay. So how it is produced also is seen here. I will not go into much detail. See, this is how it is produced. They have taken genes from daffodil. This is the name of the plant is daffodil. Daffodil has produced one gene, and Erwinia bacteria is another gene. They have taken 2 genes. 1, 2, 3 genes. They have taken 2 genes from this and 1 gene from this. They have inserted this gene into agrobacterium, this into agrobacterium, and they have infected the embryo of the rice. As a result, you get golden. We will not go into those details, right? So daffodil, beautiful color, and also Erwinia, they have taken these 2 genes and introduced them into rice plant, and rice embryo will start to produce vitamin A. This is what is happened here in case of golden rice story. So golden rice is one of the greatest contribution that scientists have made for children. I'm very specifically saying that this is for children, kids who die because of blindness, because of deficiency in vitamin A. This particular technique has really working very well in Africa, in Philippines, in Philippines we have International Rice Research Institute. The work is being done there, and this is now being used for saving kids from blindness. Very important contribution is golden rice. It took long time, but they have come up with a good product of rDNA. Okay, so these are the 5 examples that we have in our syllabus. Now, from now onwards, I will just give you the title. Okay. Yeah. Second one is salt tolerant rice. Okay, so this is the coastal region. We call these plants as mangroves. Mangroves can survive to grow in salt marshes. They have collected a gene called SOD1 gene, introduced that SOD1 gene into rice plant, and rice plant are able to grow in coastal region even if it is a salt condition. This is one good example. This is how they are now growing salt tolerant rice plant. And then next is papaya. Papaya is transgenic papaya because papaya usually gets a ring spot virus. Papaya ring spot virus is a common disease, a viral disease on this particular plant. And then scientists have used short RNAs and they have been able to overcome this. And now you have transgenic papaya plants available in the market. And then another very important thing that you can think of is your blue rose. Blue rose is not naturally available, but scientists have done have developed what you call as blue rose. They have used a gene called TFR gene from maize, and that is introduced into this plant, and then you have a blue rose. In India, we do not have much importance for cut flowers, but elsewhere, mostly in Europe, and in case of Oriental countries like Japan, there is a lot of demand for cut flowers. And rose is one such cut flower which is very, very high demand. So they have designed this particular flower, which is a blue rose, which is very important for the cut flower industry. You also have other flowers that you can see here. They have different flower colorations that you can see here. This is also done by the process of genetic engineering, right? Then drought-tolerant crops are also produced because during dry summer, arid conditions will result in death of these plants. So transgenic technology, we are able to produce drought-tolerant crops. And then this is one drought-tolerant crop in case of wheat. They have used a gene called DREB1A gene. This is control, and this is DREB1A gene. When you grow them due to drought, this will die, but this will survive. So this is drought tolerant. And then high temperature also is tolerant. Temperature plant is also tolerant. They have collected genes from this plant called Myranthus flabellifolia. Also, they have collected gene from Selaginella pteridophyta, and then they have isolated the gene, and this gene is introduced to crop plant. So we can get heat tolerant plants also. This is being worked out in case of wheat. And then we also have transgenics with amino acids. Example is potato. Okay, so in this case, you can see potato contains gene from AM1 from South American Amaranthus plant. And then potato has more protein, about 2.5% of protein is there. And also they have albumin. This is also through the process of genetic engineering. And then improved edible oil quality. They used a gene from Z maize, ZM-LEC1 gene. And as a result, the edible oil quality is improved in case of oil producing crops. And then this is a very important issue that we are now discussing with Gus Steiney. They have been able to produce male sterile lines. They have been able to produce male sterile lines. Sorry, they have been able to produce male sterile lines. We call this bar star barnase. This is one important area that they have been able to do in case of mustard, especially this is the case of mustard. And then also we have colored cotton. Colored cotton is also engineered by flavonoid biosynthetic pathway. They have taken the gene. Instead of white, you get a brown colored fibers, which actually we will not use dyeing. Dyeing is another industry which is highly polluting, both surface water as well as groundwater. So we are able to produce through genetic engineering brown cotton fibers. And then, yeah, this is the bar star barnase gene system. where we get male sterile lines produced, which is very important in breeding program, in hybridization program. I will also not go into much details of this. And then we have virus resistant plants, both in tobacco. They have been produced. Coat protein gene is one, cDNA or of RNA satellite, antisense RNA, ribosome. These technologies are being worked out to produce virus resistant plant. This is edible vaccine. Vaccines are now, hepatitis B vaccine is now produced in tobacco. It is also being suggested to be produced in potato and banana. This is also being worked out. And then we have sweeteners. Sweeteners are also produced by incorporating the gene called thaumatin into, the source is Thaumatococcus danieli. This gene is also used for producing sweetener. And then essential fatty acids are also being used, because fatty acids have become a very important issue now these days, because it is written on the oil that we use as PUFA. PUFA is polyunsaturated fatty acids. So people are not interested in high PUFA, so they want good acid, good oil for making food. So genetic engineering is working, has some work on this particular aspect. Then mineral transgenics are also available. Micronutrients, mineral transgenic iron, zinc are being incorporated into rice grain. And then transgenic banana, I think I have no photograph. Right, transgenic banana is another banana wherein a red skin banana is produced, and then you can store it for a very long period of time. Ripening genes are present there. And then you have this Barnes-Barna system. This is Barnes-Barna system. And then one very important issue that I would like to suggest at the time of closing of this lecture is one researcher at Lawrence Berkeley National Laboratory. I think, yeah, this is a lady, Professor Joanne Story from Salk Institute, California. What she is trying to do is, she is trying to tell the policy makers that carbon dioxide from the air has to be sequestered to the root system of plants, right? So as a result, see, you know, today we have greenhouse gas problem, global warming. What they say is more carbon dioxide. So more carbon dioxide, more heating of the universe, of the Earth's surface. So what she suggests is, this has to be sequestered to the air, from the air to the crop or to the natural vegetation. So as a result, what happens is, we will sequester. Sequester is holding back. We're not allowing it to go back again, holding back. This is to be held back by natural vegetation and crops. She has been working on this aspect at Salk Institute, right? What does he say? Plants suck up all the carbon dioxide, fix it, and store even roots. This concept basically splices the genes of regular crop, like bean, corn, with new compound that make them more carbon. The roots are transferred to soil and kept there. This approach essentially supercharges what the nature already does. So this is the work that is being done by this particular lady at Salk Institute, California in USA. And then the last is humulin. This is the first life-saving drug that is produced by rDNA technology. I repeat, humulin is the first product, rDNA product, produced by rDNA technology. And this is today called humulin. This is called humulin. Every house, we have diabetes problem, our elders are suffering, our younger generations are suffering. So in such a situation, through rDNA technology, it has been possible to produce this life-saving drug called humulin. I will not go into those details. You can discuss this when you ask questions, right? So this is how the technology is being worked out, and you see that in our house, our grandmother, great grandmother, grandfather will be using 1 or 2 shots of humulin. Otherwise, they will not have the sugar controlled in their bloodstream. The sugar level is either high or is low, so you have problems. So humulin is one very important compound, life-saving drug, which is produced for the benefit of elderly people who are suffering from diabetes. Okay, so the last slide is this is Bill Gates, world IT icon, and our honorable Prime Minister, Sri Narendra Modi, who are sharing that biotechnology will be the future, will bring about a lot of change in lifestyle and human life. So lastly, I thank the chief patrons today, Dr. V. Satyanarayan Sir, Principal, DDC for Women, Jagatyal, and Dr. J. Achyuta Devi, Principal, RBVRR Women's College, Hyderabad, conveners, Sri G. Chandraya, and Dr. A. Rajini Madam, and then organizing secretary, Dr. Padala Tirupati from Jagatyal Women's College. Okay, so this is a small presentation. I'll be happy to answer questions from the students. Madam, over to you.
Yes, sir. Thank you. Thank you so much, sir. Really, the presentation was awesome. And all the slides, what you are presented were apt to our subject, sir. Completely what the topic you have.
Yes, madam. Thank you, madam. Any questions I can take?
Yes, sir. From the participant side, please, the podium is open to you all. You can please ask the questions to this, sir, if any. Participants, you can mute yourself. Girls, BJT girls of RBRR Women's College. Any doubts, please, you can raise your hand or unmute yourself. Sir, there is a question from my side, sir.
Yes, madam.
So you were explaining about the flavors of a tomato, sir, where the shelf life of the fruit was increased due to the antisense gene which was inserted into the tomato fruit.
Yes.
So can I know where the gene is isolated from?
This is PG gene from any plant source, any fruit source you can isolate this. Any polygalactose gene, it can be isolated. So what they did is they have isolated this gene from tomato fruit itself.
Okay.
Okay. Once this tomato fruit starts to produce this normal mRNA,
okay,
they have immediately introduced antisense RNA.
Okay.
So antisense RNA goes and binds with the normal sense RNA. As a result, it will not produce protein at all. It will not produce the enzyme at all.
Yes, yes.
So they're actually blocking the enzyme.
Yeah.
Once the enzyme is blocked, PG enzyme is blocked, naturally the degradation of the skin of the tomato will not be there. It will take some more time.
Yes, sir. Yes, sir. So is this normal wild variety can't able to synthesize this, as you said, like from any fruit, this antisense mRNA can be introduced?
Yes, any fruit we can get, but
why can't this wild variety can synthesize its own?
No, wild variety can synthesize its own.
Normal mRNA, right?
Yes, they can synthesize. There is no problem. Okay. But in that case, the skin is very tough, so it takes longer time. But these are all cultivated and selected already. These tomatoes. See, we prefer tomato with a very thin skin, not a very thick skin.
Yes, sir. Yes, sir.
Because it is easy to cut, and then we can also they have more solids compared to the wild one. Like for example, in wild you have ramulkai. In Telugu you call it as ramul. Small. A cluster of small tomatoes are there.
Yes,
very
sir.
small tomato. If children eat them also. Am I right, Rajendra? Ramulkai tintam kada manam? Unmute chesko, Rajendra.
Mute. Okay, sir. Unmute.
Manamu ramulkai tinama?
Symptoms, sir.
It has a very small fruit, but good
Yes.
skin. So it does not get rotten very easily because PG release is also very slow. So they have isolated this, and then antisense RNA technique has been used here.
Thank you very much, sir.
Yes, ma'am, please.
Yeah. Any participant? Any queries?
Sir, I would like to ask one question.
Yeah.
In entire biotechnology or RDNA technology, okay, I think nowadays it is a complete commercial aspect is there.
Yes.
Yes. So how much it will take to reach the every farmer of the country? Okay. So and without any environmental hazard, the environmental hazard is there. Is not, or I think it is there. Environmental hazards are there due to the production or due to the cultivation of this RDNA crops or recombinant crops. But how much time it will take to reach every? Yes, in India we permitted only BT cotton only.
Yes. Yes.
Not other crops.
No. It will take a very long time in our country.
So what are the
Especially
barriers are there?
Yeah, especially fruits and vegetables. I don't think in near future they will permit the transgenic fruits and vegetable cultivation. I don't think they will do it until and unless there is a lot of pressure from the seed industry or the corporate world. It's a different thing. But for cotton purpose, because it is not a edible thing.
Edible.
So throughout the country today, at least from our standpoint of view in Telangana, all cultivation is BT cotton.
Yes.
Okay.
Even
BT brinjal also was developed by Anand Kumar. As I've told you, he's from IARA, Indian Agriculture Research Institute, Delhi. They have developed that crop. It is ready to be released, but it will be not released. But they have released this to a company, which is a seed company, which have started selling it in Bangladesh. For the last 6 years, 7 years, they have started growing in Bangladesh. So Bangladesh people are happy. They don't have a fruit borer and shoot borer problem. They're still consuming. See, we have some problems in our country. See, even transgenic technology and transgenic crops are not accepted in Europe. Europe do not cultivate transgenic crops. It is America, Brazil, Africa, and countries like ours and China. They are accepting, but not other countries. So it will take a very long time for people to pick up this technology and use it. But one very important thing I would like to suggest is there are several American companies investing in our country. For example, take your KFC. Okay, KFC, McDonald's. Okay, they are getting vegetables, fruits from their country to India. We do not know whether they are transgenics or non-transgenics. We are not very sure of it. Okay, so there is every chance of getting transgenic crops, or vegetables, fruits to our country without the knowledge of the permitting agencies in our country. We are not very sure.
I hope sir has answered to your question. Sir, thank you very
yes, madam. Okay, thank you, sir.
Okay, Rand.
Any more questions from the participants? Oh, okay, sir. Thank you very much. So now I invite Dr. Padala Tirupati, sir, to propose formal vote of thanks. Over to you, sir. Tirupati, sir.
Okay. Very good afternoon to all. It is my immense pleasure to propose vote of thanks to my godfather, Professor T.CHRISTOPHER REUBEN, sir, Professor T.CHRISTOPHER REUBEN, sir. Really, I am very happy, and because of I am the student. I am student of my godfather, Professor T.CHRISTOPHER REUBEN, sir. So, best academician, best researcher, best friend, best facilitator, best mentor, best what we can say that all types of the facilitator, my godfather, Professor T.CHRISTOPHER REUBEN, sir. So really, all the participants, we hope very enlightened regarding of today's topic. They are very happy. They felt very happy, and they are very satisfied with this lecture because of the applications of the RDNA technology is fulfill the aim of this lecture. This is not only an extension lecture. This is nothing but a national webinar also, because of the so many faculty members of various degree colleges in our Telangana state, and also other state faculty members have already joined today's your lecture, sir. So we are very happy and thankful to you for your sparing your valuable time and enlightened this more than 1.5 hour time of this wonderful lecture, sir. We are very happy and thankful to you, sir. And we are thankful to the patrons of today's national webinar, Dr. Y. Satyanarayana, sir, Young and Dynamic Principal of my Government Degree College for Women, Jagityal, and Dr. J. Achita Devi Madam Garu, Principal, the one of the prestigious institution in Twin Cities, RBVRR Women's College, Hyderabad, Dr. J. Achita Devi Madam Garu, and conveners of today's program, Dr. G. Chandraya Sir Garu, Head Department of Botany, GDCW Jagityal, and Dr. A. Rajini Madam Garu, Head Department of Botany and Food and Nutrition, and Dr. A. Kavita Madam Garu, today's facilitator, and my beloved friend, the researchers and research scholars and other faculty members of the areas of the biotechnology and also plant sciences. So thank you very much for each and everyone. We are wholeheartedly thankful to you for the grand success of this today's national webinar or extension lecture of my professor, my godfather, Professor T.CHRISTOPHER REUBEN, sir. Really, we are very hats up to you, sir. So we more expected from you, sir, because of you are the best academician, you are the role model, you are the inspiring personality forever. Till the last breath, you are my inspiring personality. We follow your complete instructions, what you have said in previously, and what you will be told me. Definitely, I will follow your instructions, sir. Thank you very much, and each and everyone. Over to you, Kavita Madam.
Okay, sir. Thank you very much, sir. Sir, thank you, sir. Christopher, sir. Hello.
Madam, thank you. Thank you so much.
Sir,
I am there. It's a privilege. It's a pleasure to.
thank you so much. You are very
Thank you so much.
accepting my request. Okay, sir. Thank you very much, sir.
Thank you, madam. Thank you so much. Bye, everybody.
Bye, sir. Dear participants, the feedback link is provided in the chat box. So please do fill the feedback link.
Thank you, sir. Thank you, ma'am.
Thank you. Thank you very much.